Hi guys, welcome to my YouTube channel. Don't forget to subscribe and share.
Hi, Assalamualaikum warahmatullahi wabarakatuh. Okay, in this video, I'm going to explain for subtopic 1.1, 1.2, and 1.3. Okay, let's start. Okay, actually, biodiversity is very interesting chapter because we're going to know how the species in our universe is being classified. So, ini kerja yang sangat menarik untuk kelaskan spesies, meletakkan spesies itu dalam kingdom, dalam kelas dan sebagainya. Okey, untuk subtopik 1.1, apa yang kamu kena tahu is three types of biodiversity. Okay, the first one is genetic diversity. As you can see, genetic diversity is a variation of all living form at the genetic level. Variety of genes, allele or nucleic acid. For example, okay, katakan saya ada rambut yang hitam, okay, dan Ali pun ada rambut hitam. Okay, jadi secara phenotype-nya kita nampak sama, iaitu rambut hitam. But then, maybe they have a genetic variety which my hair, the genotype is B besar. But then Ali tadi, the genotype is B besar, B kecil. So, itu dari segi susunan allele dan berbeza dari segi genotype. So, itulah menunjukkan genetic diversity. Okay, next, kita ada species diversity. So, species diversity is refer to the variety of species or needs in the number of the species in a community. So, as you can see, kamu nampak sendiri, dalam dunia kita ada macam-macam spesies. Ada manusia, ada orang hutan, ada monyet, ada unka dan sebagainya. So, kalau kita cerita tentang harimau sekalipun, okay, harimau, the genus is Panthera. Panthera ada macam-macam spesies. Ada Panthera pardus, Panthera tigris. So, menunjukkan the species adalah sangat banyak, okay, dan berbeza. And then we have ecosystem diversity. So, ecosystem diversity is variety of ecosystem that are present in the biosphere. So, for example, kita ada lake ecosystem. Okay, bila kata lake ecosystem, dia punya abiotic component mempengaruhi biotic component. Contohnya, we know that lake ecosystem dia adalah fresh water, iaitu air tawar. So, bila air tawar, ikan yang ada dalamnya hanyalah ikan yang boleh survive, okay, pada fresh water. For example, tilapia, lampam. Okay, but then if we talk about marine ecosystem, the water is salty. So, the salty water is not suitable for tilapia and for the lampam to live. Okay, so you can see that different ecosystem. So, that show ecosystem diversity. Okay, next is hierarchical classification. For hierarchical classification, this is proposed by Carlos Linnaeus. Okay, in hierarchical classification have 7 taxa. Start with domain, itu yang paling besar. Domain, kingdom, class, order, family, genus, species. Wajib ingat ikut susunan. Paling besar is domain, and then yang paling kecil itu adalah species. Okay, and each of these we call as taxa. Okay, kingdom is taxa. So, if the question asks you what is taxon of X, okay, so the answer is class. Right, next is type of classification system. We have 2 types of classification system. First is based on 5 kingdom system that proposed by Whittaker. Okay, and second one is 3 domain system which proposed by Carlos Linnaeus. So, in 5 kingdom system, the kingdom is kingdom Monera, Protista, Fungi, Plantae, Animalia. Wajib ingat ikut susunan. Wajib ingat ikut susunan. Why? Because the sequence start from Monera until Animalia. This is refer on the complexity of the organism under the kingdom. So, the simple organism will be classed and the kingdom Monera. And then the organism become more complex sampailah dikelaskan ke dalam kingdom Animalia. And then we need to know how do we take a classified organism under this 5 kingdom system. Okay, dia gunakan tiga komponen. Okay, the first one is according to level of organization. Either the organism is prokaryote or eukaryote. So bila banyak spesies yang ada, so dia kelaskan dulu. Okay, mana-mana spesies yang prokaryote, dia kelaskan itu sebagai kingdom Monera. So bila kata prokaryote, eukaryote, sepatutnya you don't have to be confused, because you learn about prokaryote, eukaryote since your secondary school, and then kita bagi peneguhan lagi untuk chapter 2 last semester. So when we talk about prokaryote, the main characteristic of prokaryote organism, they are unicellular. They do not have true nucleus, and they do not have membrane-bound organelle. So if the species show this characteristic, they will group under kingdom Monera. So for the rest, okay, for the left species means they are eukaryote, and then if they are eukaryote, so second element to be considered is level of organism. Either the organism is unicellular or multicellular. Yes, all the rest yang tinggal sekarang ni hanyalah eukaryote. And then kita nak tengok pula, if the organism is unicellular, so we will classify the organism under kingdom Protista. Okay, eukaryote, unicellular. So they will belong under kingdom Protista. And then for the rest, okay, the third element to be considered is mode of nutrition. Okay, either the organism is saprophytic, photosynthetic, or holozoite. Saprophytic organism is the organism that absorb the nutrient from that organic matter, and then for photosynthetic organism, the organism produce their own food. And then for holozoite is organism that digest and absorb nutrient. Okay, so for fungi, for saprophytic organism, they will be grouped under kingdom fungi. For photosynthetic organism, they may be grouped under kingdom Plantae, and for holozoite, they may group under kingdom Animalia. Alright, so if the question asks you, state 2 unique characteristics of kingdom Plantae, can you give the answer? The answer is simple as Plantae are photosynthetic organism. Plantae is multicellular. And for kingdom Plantae, the organism is eukaryote. Bukannya 2, kita boleh bagi 3. So this is very important to know the basic classification of species under 5 kingdom system. Okay, now let's proceed to 3 domain system that proposed by the Carl Woese. So 3 domain system, the 3 domain is domain Archea, domain bacteria, domain eukarya. Okay, compared to the 5 kingdom system, okay, all prokaryote is being classified under kingdom Monera. But in 3 domain system, the prokaryote itself will be divided into 2 domain, which is domain bacteria and domain Archea. So that's why you can see here, for bacteria and Archea, both are prokaryote. So for eukaryote organism, they may be grouped under domain eukarya. So under this domain eukarya, so have 4 kingdom, which is kingdom Protista, fungi, Plantae, Animalia. Okay. So settle for subtopic 1.1. For subtopic 1.2, okay, we're going to focus on domain bacteria and domain Archea. So in terms of 3 domain classification, okay, domain Archea and domain bacteria, they are actually different domain lah. But when we talk about 5 kingdom system, these both, the bacteria and the archaea, they belong under kingdom Monera. So the basic thing about kingdom Monera, about the dormant archaea and bacteria, they are prokaryote. So please remember at least 5 unique characteristic of prokaryote. Tadi saya dah bagi 3, simple je kan? They do not have true nucleus. They do not have membrane-bound organelle. They are unicellular. So you can add another 2. Apa yang mudah untuk kamu ingat? Kamu boleh ingat macam-macam. The ribosome is small, which is 70 unit. The DNA is sliced freely in the cytoplasm. Then the DNA is in circular shape. A lot of other characteristic of prokaryote that you can remember. Okay. So what you should know is the difference between bacteria and archaea in terms of cell wall, structure of lipid in plasma membrane, association of histone protein with DNA, and response to antibiotics, and also the example for each domain. Okay, in terms of cell wall, bacteria, the cell wall may have peptidoc lichen. But for archaea, they do not have peptidoc lichen layer in the cell wall. Then in terms of lipid in plasma membrane, so for bacteria, they have unbranched fatty acid. Compared to archaea, some will have branched fatty acid in the membrane. In terms of association of histone protein with DNA, for bacteria, the DNA will not associate with histone protein. But for archaea, the DNA associate with histone protein. And in terms of response to antibiotic, for bacteria, the growth is inhibited. Okay, but for archaea, the growth is not inhibited. Maksudnya, bacteria, dia punya proses growth itu boleh direncatkan dengan adanya antibiotik. But for archaea, dia tak kisah, dia masih boleh survive. Okay, walaupun dalam antibiotik. So example for bacteria is E. coli, Escherichia coli, and for archaea is Sulfolobus. Suka saya ingatkan dekat kamu, penulisan saintifik kamu kena titik beratkan. For example, this E. coli, this E is referred to Escherichia. Okay, the first word is referred to genus name. And the second word is referred to species name. So for genus mestilah huruf besar. And then for species dimulakan dengan huruf kecil. And then for Sulfolobus, kenapa kita nampak hanya satu patah perkataan dan diikuti dengan species? So this huruf besar, betul tak? So this Sulfolobus is referred to the genus name. And it not give you a specific scientific name. So that's why dia letakkan species. So, menunjukkan spesies ada genus Sulfolobus. Okay. And make sure setiap kali kamu menulis scientific writing, the technical is very important. First word is genus name, huruf besar, mesti digariskan perkataan itu. And then the second word is refer to species. So, mulakan dengan huruf kecil dan wajib digariskan. Dan sekiranya we don't have the species name, okay, we just have the genus name, sama juga perkataan genus itu mesti digariskan. Okay, dan perlu ditulis SP kat belakang, refer kepada species. Alright, so for subtopic 1.3, diversity of bacteria. So for domain bacteria, okay, we can classify the bacteria into these 3 diversity. First, according to the cell shape. Maksudnya dekat sini, dalam domain bacteria ni ada banyak jenis bacteria. So kita pecahkan pula, kita kelaskan pula bakteria-bakteria ini mengikut diversity, mengikut jenis-jenis tertentu pula. So first, kita tengok according to the cell shape. So, bakteria, dia akan ada pada 3 bentuk utama. 3 bentuk utama saya cakap. Maksudnya dia boleh ada dalam bentuk lain lah yang kita tak sebut kat sini. Contohnya vibro. Vibro dia macam bentuk vibrate. Lepas tu dia ada bentuk yang macam bentuk koma. Tapi kita tak nyatakan kat sini. Silibus kamu kita tekankan only 3 shape. The first is coccus. So bila kita nampak coccus word, that is refer to spherical shape. So kalau bakteria itu bentuknya bulat, so kita panggil itu sebagai coccus. And then kita ada bacillus. Bacillus is refer to rod-shaped bacteria. So kalau bentuk panjang-panjang, kita nampak macam bentuk panjang macam ni. So ini is refer to bacillus. And then kita ada spirillum. So dia bentuk spiral. So sebenarnya ada lagi jenis vibro. Okay, tapi kita fokus kepada 3 ini. And actually this can arrange for coccus especially. Dia ada dia punya arrangement. Kalau dia dalam bentuk double, kita panggil diplococcus. Kalau dia dalam bentuk chain, kita panggil dia sebagai streptococcus. Dan kalau dia dalam bentuk cluster, staphylococcus. Okay, so itu adalah cara arrangement dia. Next, according to Gram stain. So dalam Gram stain, kita ada 2 bacteria, 2 groups of bacteria, which is gram-positive bacteria and gram-negative bacteria. So this gram-positive and gram-negative bacteria we can identify through gram staining method, which is experiment 7. So during that experiment, kita akan stain the bacteria using crystal violet, and at the end we're going to see the appearance of the bacteria colony under microscope. Either the appearance is violet color or purple color or pink color. Okay, so if the appearance is dark purple or violet color, okay, that is referred to gram-positive bacteria. Okay, but if the appearance of bacteria in pink color or red color, so the bacteria considered as gram-negative bacteria. So for this gram-positive and gram-negative bacteria, you should know the difference in terms of cell wall, crystal violet, cell appearance, and resistance toward lysozyme, and of course the example. So in terms of cell wall, since they are in the same domain, which is domain bacteria, so both will have peptidoglycan layer, but the difference is in terms of the thickness. For gram-positive, the peptidoglycan layer is thick. Gram-negative, the peptidoglycan layer is thinner layer. So when we drop a crystal violet, what happens is in gram-positive bacteria, the crystal violet is trapped in the cytoplasm because dia ada thick peptidoglycan layer. Jadi bila kita rinse out the crystal violet tu tak boleh nak keluar untuk dicuci sepenuhnya sebab dia terperangkap dalam layer peptidoglycan itu. Okay, tapi compared to gram negative, peptidoglycan layer dia nipis. Jadi bila kita drop crystal violet and then kita rinse out, apa yang jadi? The crystal violet is easily rinsed from the cytoplasm. So the consequence is the appearance. So that's why gram-positive bacteria will appear dark violet or purple, compared to gram-negative will appear pink or red in gram staining. So in terms of resistance to lysozyme, so gram-positive bacteria, the peptidoglycan can easily be digested by lysozyme. So it means that this type of bacteria can be killed easily lah. Compared to gram-negative, the peptidoglycan layer with outer membrane contain lipopolysaccharide layer. So this lipopolysaccharide layer is actually extra defense barrier for the bacteria. Okay, katakan kita dah tahu bakteria dia ada kapsul. Okay, semua bakteria ada kapsul. Kebanyakan bakteria memang ada kapsul. So untuk gram negatif ini dia akan ada satu layer extra pada kapsul itu. So sekiranya ada lipopolysaccharide layer tu, ada extra layer, so bakteria ini lebih susah sedikit untuk dibunuh. So kalau kita cerita dari segi harmful, gram positif, gram negatif, both still harmful lah. But then the most harmful is gram negative. Why? Sebab dia lebih susah untuk to be killed ataupun to be digest by the lysozyme. So for the example, for gram positive is Staphylococcus. Okay, and then for gram negative is Escherichia coli. Or you can simply say E. coli. Okay, and the third thing for bacterial diversity is in terms of position of flagella. Okay, so position of flagella, look at the diagram for monotricious. So what we can interpret from the diagram, the bacteria will have a single flagella at one end. Lipotricious, the bacteria will have more than one flagella at one end. For ampitricious, kita boleh nampak the bacteria will have the flagella, okay, at both ends. And then for the peritricious, the bacteria will have the flagella entire of the cell. Bukannya di hujung sahaja, tapi di keseluruhan permukaan. Okay, so the last thing is about the importance of bacteria. First is recycling of chemical element in ecosystem. So we know that bacteria memang digunakan untuk, contohnya dalam carbon fixation, lepas tu nitrogen fixation. So kita ada nitrobacter. So itu semua adalah bakteria yang terlibat dalam recycling of chemical element in ecosystem. Then for symbiotic ni contohnya kita sendiri, okay, antara our body, the human with the E. coli. So kita pun tahu kita memerlukan E. coli to digest the lactose. So that is in term of symbiotic relationship lah. And then pathogenic, of course, okay, most of bacteria is a pathogen. Maksudnya dia boleh menyebabkan harmful, dia boleh menyebabkan disease. For example, kalau the bacteria is syphilis, okay, dia boleh menyebabkan penyakit syphilis lah, Corella, and then bacteria rubella. So the bacteria can cause harmful to human and will cause disease lah. And then research and technology, we know that E. coli, for example, is used as host cell in DNA cloning technology. So obviously it will be used in research and technology. So that's all for subtopic 1.1, 1.2 and 1.3. See you in the next video. Bye.